Professor Wanda Markotter holds a PhD in virology, which she obtained after starting her academic career at University of Pretoria in 2004. Now she is the director of the Center for Viral Zoonoses, Department of Medical Virology, Faculty of Health Sciences at University of Pretoria. She occupies 2 research chairs, the DSI NRF South African Research Chair in Infectious Diseases of Animals, Zoonoses, and a chair in People, Health, and Places at the Future Africa Institute. Her interdisciplinary research program focuses on bat pathogens and the risk of spillover in Africa. In 2021, she was appointed as the co-chair of the One Health high-level expert panel, advising the WHO, OIE, FAO, and UNEP. Professor Wanda Markotter's talk today is entitled An Interdisciplinary Approach to Biosurveillance and Understanding Spillover from Bats. Thank you very much for joining us, and please take it away.
Thank you, and afternoon to everybody. It's evening in South Africa, but a warm welcome to everyone. So I hope I'm still awake and full of energy for this talk, but I'm going to try. It's at least not that late, but thank you for the opportunity. And I'm going to start with really giving you an overview of the important literature, just a snapshot to set the stage. But then I'm really going to go into what we've been doing in South Africa and share some of the research that we've been doing. I'm going to try and not give too much detail, but you're welcome to ask more in-depth questions afterwards. So really thank you for the opportunity, and it's really nice to join everyone on this afternoon. So to start with, let me just see why my things are there. So to start with, just where does spillover fit in? And, you know, we hear all these words about prevention, preparedness, response, but where does spillover really fit into this picture? And this is a very nice paper that came out recently by Bernstein et al. That had quite a nice diagram that I like, that shows your pre-emergence phase where you have viruses circulating in wild animals. And this can be different wild animals. I mean, I'm going to talk about bats today, but the picture is actually much broader than bats. It's not just bats. And then you have your pathogen spillover. And if you look at these arrows, it can happen directly to humans, but that happens actually quite seldom. You know, normally there's some kind of intermediate host, or some people call it an amplifying host. There's different terminology for these things. But it's basically something in between where the virus spill over from wildlife into humans, and it actually becomes it's amplified to a higher titer in these animals, and that's why it's easier to spill over. And then there's also your vector-borne that I'm not going to talk about today, where your ticks and your midges and your mosquitoes are involved, and they serve as a vector. And what's quite important about this little block is there needs to be contact. You know, these things that we know is in wild animals, and it's just always there. That's where they are. That's where they evolve. So we sometimes talk about the evolution of these pathogens in these wildlife hosts. They've been there forever, not new. They've always been there. There's a reason why we get to the stage where we have emergence in humans. And this has got to do with contact due to several reasons. And it's things like deforestation, wildlife trade, urbanization, all your land use changes. It's human behavior causing a lot of these issues. And then we've got emergence in humans. And we've got this first phase where if we do really very good syndromic surveillance, because what we need to understand, if the pathogens are in wildlife, we're not seeing disease. These animals are used to dealing with these pathogens. So we're not seeing disease, but if it spills over to humans, we're actually seeing disease. And that's why we start talking about syndromic surveillance. So we will look for certain symptoms. Is it encephalitic? Is it fever, febrile illnesses? And if we have very good surveillance, we will detect it easy in humans. But that's unfortunately not always the case. So we will see through our syndromic surveillance, something has spilled over. What we usually start seeing is when we have more than one human case. And then we can still contain it localized in a small area. We've seen this a lot with Phyloviruses, where we have spillover of Ebola in a small area, we contain it immediately and it stops there. But in some cases, it becomes an epidemic where it spreads in that area. And then pandemics, I think we're so used to, or we all know now what a pandemic is after COVID-19. I mean, it just spreads due to global travel. We just all connected, and a disease that starts in West Africa can the next moment be in New York and cause a pandemic, as we've seen with COVID. So where my research fits in is really in this pre-emergence pathogen spillover emergence interface. It's really about prevention. It's not when the disease is already in the human population and established in the human population. So just to make it clear where we are in this picture. And why do we want to look at that early phase? And there's quite a few papers out. This is again a recent paper. It's from the Bernstein paper, where people are looking at reasons why we should look at this pre-emergence phase, at the spillover phase. Why should we look at wild type? You know, what is the advantage? You know, shouldn't we just react when it's in humans? Shouldn't we put all our resources there? And there's clear advantages to prevention in terms of economics. There's quite a few papers out on it. This is like I said, one of the recent papers that shows we're going to see more epidemics. We're going to see more pandemics. It's increasing. It's spreading. If you look at these little dots at the bottom graph, the dots are becoming broader, showing there's more countries involved. There's more people dying from these diseases that's emerging from animal origin. And then this is just a table showing some of the bigger events. A lot of them has been associated with influenza, the Spanish flu, and then H3N2. You'll see there was quite a lot of deaths. HIV is still very much in the picture. That's still something we're still dealing with. And then COVID-19 is significant in this picture. It's not an insignificant pandemic. I think we all know that with all the implications. And if we really start looking at the economics, and this study specifically used different parameters than some of the other economic studies, so it's not exactly comparable because they use different analysis, but it shows that if you just invest to react when something is already there, it will cost you really a lot of money. And if you look at this, for instance, it's between 35 billion to 21 trillion. If you just go for a situation where you just want to try and limit outbreaks, you know, maybe 10% less, because everybody always tells us we can't, you know, we can't predict everything. There's going to be some things that's actually going to happen even if we try and prevent. It gets down to about 32 to 19 trillion. But then if we can cut events up to half of it, we're actually getting down to saving about 1.6 million lives and $10 trillion in that process. So that is one of the studies really saying that we should invest in prevention. If you look at the diagram on the other side, this is all the costs in terms of preventative measures. So it looks really, I mean, if you tell a country they're going to have to invest in this amount, they're going to be what? I'm not going to invest that amount of money, especially in the developing countries. They don't have that money. But if you start looking at the advantages of investing that amount of money versus damages of COVID, and this was already published in 2020, these amounts I think is much higher now in 2022. And even other things that's really important, like environment and climate change, if you start investing in some of these prevention activities, you actually also have other spin-offs in terms of carbon emission, in terms of climate change, and that's also got a value in terms of money that we need to consider. So we really need to start looking at the bigger picture, which is really a shift of thinking. It's not just an immediate reaction, but in the long term, there's clear benefits to start looking really at prevention and not just reaction. So that's just to set the stage of why we're doing it, because I always get this question, why are we looking at viruses in bats, you know? And there's clear reasons for that. It's not just about the viruses, and I will get to that point, but it's a bit broader, but there's clear benefits. And why bats? So there's been several studies where people tried to motivate why we should look at bats. One of the first studies by Lewis et al. Did a study looking at rodents versus bats, and does bats really have more viruses than rodents? And they accounted for sampling bias and all kinds of things using really clever statistical methods, and bats came out on top. Then there were some other studies after that that re-looked at this in a more holistic approach. And yes, bats are still important. Bats are one of the taxa that we need to look at, but it's not necessarily the only one. And I think after the first SARS epidemic that happened in 2002, we basically shifted all our focus to bats, and we started forgetting about some of the other wildlife. And I think we need to get back to the point where bats is important, and it's important in terms of potential zoonotic diseases, but it's really not the only animal in that picture and the only factor in that picture. So if you look at this study by Melenster and Stryker, they really identified mammals are important in terms of this whole picture, looking at potential zoonotic viruses, accounting for the amount of diversity in these groups, as well as the amount of surveillance. Bats, rodents, and primates are actually all important. And then the birds, I mean, we can think with avian influenza, that's always an important host to look at. So that's what's coming out on top in terms of what we think we need to look at if we look at modeling studies and prediction studies. So I think it's quite clear that we need to look at bats. I don't think we can ignore them, but we need to also look at a bit of a broader picture. And if you look at this paper, it's also from Plowright et al. It was a review. There's several factors that we need to actually consider if we look at wildlife spillover. And I think this summarizes it quite well. You can't just say, I'm going to go out and I'm going to test every animal just for pathogens, and then I'm going to say there's going to be a zoonotic potential. The picture is much broader. Like I showed at the beginning, there needs to be contact. So wildlife host distribution and density becomes quite important. This is basic data that we actually need from the host that a lot of the virology studies are not addressing. You know, what is the area that this host are actually distributed in, the density, and how's things like land use changes, habitat fragmentation going to change that? Because if we find a virus, for instance, in a forest where there's no contact with humans, if we leave that forest alone, what is the risk? You know, so if we go into those areas and we test and we find a Ebola-like virus, but there's no contact with humans or there's no pressure on that environment, that's probably a relatively low risk. So we really need to direct our efforts where there's going to be risk and understand viral diversity in those areas, combined with some of the other factors. And then you've got a whole issue with host susceptibility that's also got to do with natural resources and the condition that the animals are going to be in. Even things like pollutants and pesticides influence these things. We started to look at it in South Africa in bats, where we look at the influence of pesticides on bat health, and immediately you see an increase in viral diversity. You know, so there's so many factors affecting this ecosystem that can play a role in spillover of diseases. Then we need to know where the pathogen is excreted. And retrospectively, if we look at the Africa studies, in filovirus studies, we used to go and do destructive sampling and then look for a virus in the liver. And we would say, oh, we find a Ebola virus or a Marburg virus, but we didn't really in the past really study where the pathogen will occur outside of those and what is the survival of that pathogen. You know, is it in the urine? Is it in the fecal? Is it in the saliva? Because how else is it going to get to an intermediate host or to humans? And then a very, very important part that I think has been also highlighted by COVID is human behavior. That contact really needs to be there. If that's not there, and this is several factors, it's very, you know, we make these lists, and I will also highlight it later, of factors that can lead to spillover, but it's general factors. And we're not really understanding on a regional or a community level what the actual factors are. And we use global predictions to say it's going to be wildlife trade, it's going to be bushmeat hunting, it's going to be urbanization. But do we really understand what's going on the ground? Because that's the only way that we will really put in mitigation actions that will really work. And then we've got human susceptibility. We can have things like pre-existing conditions, decreased immune function, all kinds of things that can lead to increased susceptibility in humans. And then also human actions, that things like crowding, movement of people or products or services. So this is just to give you an idea of how complex this is. You know, it's not just about detecting the pathogen. There's so many other factors that needs to be looked at. And this has been summarized in several studies and how important people think it is. You know, bushmeat, food industry changes, human behavior, climate is always part of it. Climate has got several effects. You know, it can have an effect on movement of animals, or it can have an effect on the health of animals.
You need help carrying it?
Yeah, these two things.
And then there's all kinds of land use changes that can also play a role. And people have indicated percentages of importance to these things. That's quite, it's global. It's not really on a regional level. So that's one of our issues when we look at interdisciplinary spillover investigations of BAT pathogens, that we don't really have this detail on community or regional level. We're making predictions based on global data. So getting to bats, so I don't know how much you know about bats, but bats are the second largest group of mammals after the rodents, so extremely diverse. There's lots of families. You can just see all the faces of the bats that you can see. They look different. They occupy different ecological niches. So it's really a complicated group of animals. Some of them live in caves, some of them don't. So you can't just say that the risk factors for one bat can be extrapolated to other bats in terms of viruses. You really need to look at the ecology of these animals and understand all the biological and ecological factors. If we get to Africa, we also have a lot of species in Africa, about 320 species. If we go down to South Africa, we're looking at about 60 species. It keeps on getting more with genetics these days as we split them. So it's actually increasing. And we have a large diversity. What we have in Africa, we've got the fruit-eating and the insectivorous bats, if we divide them in two broad groups. We don't have vampire bats. We're at least not dealing with that in Africa, but we have the ones that eat insects and the ones that eat fruit, belonging to several of these families. So recently, or it's not so recent anymore, it's been, I think, just before COVID, we wrote a review on what we know about what we would call the high consequence zoonotic pathogens. It's always these groups, the coronas, the phylos, and the Paramyxoviruses. The coronaviruses, I think we all know quite well now. There's the SARS groups, and there's the MERS, and then there's also others that cause the general cold, like a 229E and NL63. And then with your filoviruses, it's Ebola and Marburg. That's quite well known, with quite a bigger diversity recently identified that we don't even know if it's involved in human spillover. And then the Paramyxoviruses, the ones that people typically know is Nipah, where we haven't seen any human outbreaks in Africa, but it's associated with other regions of the world. And then your orto and your pararabula, where you have measles related and mump related, and we even have Suga virus in Africa that actually spilled over to one biologist that was working with bats. So this is sort of the picture that we know. And if you look at these maps, the dots are implicated bat species and where they occur. The shaded regions is where we actually have virology data. So what I wanted to illustrate with this map is we have very limited data in Africa on virus presence and diversity actually for these important groups. Very sporadic studies in countries, but if we look at the distribution of the bat species, it's much broader. So we would expect to see it in a broader range of countries. Marburg is a very good example where we recently had a case in Guinea. And it was the first case. Why? Because the bat is occurring there. It's actually not that surprising. These viruses are quite bat specific, bat species specific. So we really need to understand the diversity of our landscape in terms of pathogen prevalence and where it occurs in terms of potential hosts for viruses that we know can be zoonotic, but there will always be the ones. I mean, we didn't see SARS coronavirus 2 coming. So we also need to understand the full diversity and try and make conclusions based on that and risk mitigations if necessary. So it's not all just about pathogens. So yes, we need to look at what's going on in the pathogens, but that's a very small part of the picture. The other part that we also focusing a lot on, and we've done quite a lot on this, is really understanding the host. So this is the ancient and co-evolution with the pathogens. It's the life history of the host. It's how they move, because movement tells you if there's going to be contact with people. And it's not so easy to track bats because they're not so big. So the technology is not always so easy to do, but we're trying to do that. Is there any resistance? When is the shedding? Is there any tolerance? How does the social structure look and their dispersal? The age, sex, and reproduction strategies became quite important for us in South Africa because we could link these factors to actually higher excretion levels, which immediately tells you maybe when your risk periods are. So this is something that you actually need to understand. And the only way that you can understand this is by longitudinal studies, which means you need to do a lot of sampling over time. You can't just go into a country, sample a lot of bats and go out and say, this is what we know in terms of how these viruses are maintained and how persistent they are in the natural reservoir. And then the other thing is all about human and animal interfaces. While you're doing this, you also need to consider all these other things. You know, what is the anthropogenic factors and what is the risk factors that you really need to look at? Then spillover hosts is also really important, and there's extremely limited studies in Africa in terms of this. It's always been focused on the wildlife host, and we don't really have evidence that it's actually spilling over to other species using serology in humans or other animals that have close contact with bats. And it's been due to technologies also that we don't have for these diverse pathogens, but I think we're now at the point where we actually need to look at this. And although my work has been very much focused on bats and understanding the reservoir host, we're now planning from the beginning phase to also implement some of these other studies. But we don't have all the results for that yet because it's quite long term. But we're implementing and trying to do that in parallel with looking at the bats. So we really do longitudinal sampling sites. We're in a good situation where we're in country. We focus on Southern Africa, and we've identified sentinel sites. It's just a few pictures, but it's really based on sites that's in the middle of our cities. This cave is in the middle of the Johannesburg, Pretoria area with quite high risk species in it. So it's really in an area where there's contact with humans. This is in an urban area, a rural area where people are having subsistence farming and everything and fruit trees around the area. And this is both these two caves, Gatkop and Matlapitsi, is used for religious purposes. So in South Africa, a lot of our religious groups still use caves, and they go in there, they have their ceremonies, even circumcision. So it's really high risk areas that we've identified, and we're doing sentinel surveillance to try and identify what pathogens are present and high risk activities. This is just a few pictures. So we decided a few years ago that we will only do non-destructive sampling. So for specifically the reasons I've explained at the beginning, we need to know where the viruses are excreted. To tell me a virus is in a liver of a bat, yes, I've got a virus, but what is the real risk if I don't know where it's excreted? So we really look at swabs, urine, fecal, identify species based on genetics, and then we take blood to use in serology. One of the things we also started with quite a few years ago is tattooing bats, and we're using normal human tattoo guns. so we give them a nice number, and we could track them over time in terms of recaptures. So we could look at viral presence over time, but we can also look at serology over time and where they move between caves. So this is also some of the data that we're collecting. One of the additional things that we can do that I'm not really going to talk a lot about today is because we've got high biocontainment facilities, we have access to biosafety level 3 and 4 labs and an experimental bat colony. We do sometimes do experimental infections to really look in controlled environment how the virus is persisting in the bats and how it is excreted. So we've done some filovirus work specifically together with National Institute for Communicable Diseases. So it's really looking at this full picture. And then I just wanted to share a few examples. I don't think I need to tell anybody about coronaviruses and how diverse they are. But yes, there's different hosts with coronaviruses. We've seen human coronaviruses throughout the years with some of the significant outbreaks, MERS in 2012, then SARS coronavirus 1 a bit later, and then we saw SARS-CoV-2 that's now causing real havoc. So the coronaviruses have always actually been sort of a red flag. We should have really watched them after the previous outbreaks because there's been proof that they can spill over, and they can spill over from very different hosts, with bats hosting a very, very high diversity and probably co-evolving with some of these viruses. So we again, as we focus on Africa, wrote quite a extensive review a few years ago where we really looked at what was done in Africa in terms of bats, and specifically also looking at the SARS-related viruses. So I use the word related because we don't find SARS-CoV-2 in Africa, but we find related viruses in Africa. And related can mean a lot of things. It can be very distant, but it still falls into that group in a phylogenetic tree. So we need to be careful how we use that word. It's really not the same virus. And what we found is, again, looking at countries, so the darker the colors is the amount of publications. Several countries, we have absolutely no data. And some countries, it's one-off studies from one area where it was very opportunistic. South Africa, we've got a few. But a lot of countries, we know really very little. And if we look at MERS and 229E and NL63 and SARS coronavirus 2, at this point when we published this, there was only 2 countries reporting SARS-related viruses. And this is now, we use this data and say, well, there's no SARS-related viruses in Africa. But we need to look at what we actually have done in Africa. We've done very little. We're not understanding the full extent of diversity of SARS-related viruses in Africa. And if we look in more detail, and this is all in our published studies, what we really identified is how little rhinolophus, which is the horseshoe bat associated with SARS-related viruses, we actually sampled in Africa. Very, very few. So our surveillance was just not adequate. And I can tell you from the work that we've done, and I'll show it also now, when we started doing increased surveillance, specifically focusing on these high-risk families, we did find SARS-related viruses in South Africa. So it's sometimes a surveillance issue on what we're sampling. If we look at the second table, this is what I mentioned at the beginning about spillover hosts. We've done almost nothing on spillover hosts. So if you start looking at the amounts here at the bottom, it looks like we've actually done a lot, thousands. But if you go and look, almost 90% of that was one study on primates in an enclosure. So it's not really looking at the full diversity of potential spillover hosts. So we're really not understanding that picture and what's the potential of spillover in Africa. So we do biosurveillance using your normal PCR, RT-PCR assays, really looking at small regions of the conserved region of the genome and looking at excretion. So directly from bats, but also population level, where you put sheets under colonies and you try and get just a broader idea of coronavirus diversity. And this is what you get in all studies, is these crazy phylogenetic trees with diversity all over. So trying to, it's not different in any part of the world. You can now try and, say that one's important or that one's important. The red ones are human ones that we know of, that we know spilled over or as human viruses. And then the rest is just this crazy diversity that we find in bats. So it's really difficult from this to decide which one is going to cause the next outbreak. So you can't really actually make inferences. You can say they're closely related to human viruses, but it's not based on receptor binding domains or any characterization. It's just based on a phylogenetic tree. And that's where the bigger picture becomes very important, because can we really single out one of these viruses, or do we need to look, yes, we've got diversity, it's not good. This diversity is in bats. It can recombine in the case of coronaviruses. It mutates at quite a high rate, which is not good. So if we have contact of all this diversity with humans or with other animals, at some point, something's going to happen. And we really need to address those factors. So if we start looking at some of these clades, we know they're very bat specific. And this is some of the work that we've now done in South Africa, where we find related viruses to SARS coronavirus 2 in bats in South Africa. So we know there's some, they're very related, they're still very different, but they belong to these clades. So that's a red flag. We don't really want this interface in these areas where they have the potential to spill over and they have this potential regularly. What's also quite interesting, and this is more on an academic side, because this is not at all a potential zoonotic virus, although these days I'm not sure if we can really say that. So in our fruit bat populations, we sampled really over time. That must be 2017, not 2017. But we sampled over time and we get this huge diversity. But what was really interesting is if we look at this diversity over time, they peak at different times, which just complicates the situation even more. So the viruses that we're seeing in fruit bat populations is not excreting in fecal material at the same time. So depending on what variant you're looking at in this picture, some of them may peak in March, some of them may peak at another time, some of them may peak in July, which actually makes it's much more complicated in terms of risk. But this is really understanding the viral persistence and maintenance in bat populations. And what we're trying to do with this data is really looking at risk in terms of if people enter these caves and when there's contact with humans. So what we've also done, I need to speak faster now. What we've also done is combined this with the reproduction season. So we typically getting peaks when females are pregnant, not so high. But what we see, and this is typically with a lot of other studies, is you get all these juveniles into the population that's got maternal antibodies for a while. So they're protected because their mothers had antibodies. But then if that wanes, you suddenly get these incretion of viral excretion in fecal material. So it's all to do with the reproductive season. You know, when these animals are losing their maternal antibodies and they have this period where they're susceptible to virus infection and they're actually shedding it into the environment. With the Paramyxoviruses, we're seeing a very similar pattern. Also, excretion peaks only certain times of the year, where if we look at this over 2 years, there's times where there's just no virus in the urine or in the fecal material. But then there's times where there's all these peaks where you can connect it again to the reproduction season. We do know that we have the Nipah-related viruses. So this is part of the Nipah Hendra clade. It's not Nipah, it's not Hendra, it's still very different. But we have some viruses that falls into these clades if we put them in phylogenetic trees. So we need to watch these, and they're typically associated with urine excretion and then respiratory illness in people if they do spill over. And again, seeing these excretion peaks in the winter months, but again, an excretion peak in spring. And it's again got to do with what we think the maternal immunity of what we will call the sub-adults or the juvenile bats. And also then the recolonization, because the bats move around and then they come back, and the late pregnancy phase. So we're trying to really unpack, you know, where are these risk periods? And this has been mostly done on our fruit bat species. And then with the filoviruses, we have Marburg in South Africa. In our bats, we haven't seen any spillover into humans. So in other areas in Africa, there's been spillover into humans, and we're really trying to see if this is due to human behavior factors, because the virus falls in the same group. I don't think there's a pathogenicity issue. It's really to do with human behavior. We have it in our fruit bats, but we just never have seen any human outbreaks. which is probably due to the spillover factors like bushmeat hunting, land use changes, and contact with our fruit bats. And when we look at some of the experimental infections, I'm not going to go into detail on this, we know these viruses are excreted in urine. We know we even get saliva swabs positive sometimes. We do see it in fecal. So that risk of transmission is there. We're not sure how it's maintained in bats. That's still a very, very vague answer. But we do know we see the virus in some of these excretions that could lead to spillover in humans. Some of the other things we've been doing, and this is where the interdisciplinarity comes in, is really looking at age. And I think if you listen to some of the previous graphs that I showed, age is becoming really important. We want to be able to tell you a bat is 3 months old, the bat is 5 months old, the bat is 7 months old, and it's not so easy. We can only say sub-adults, adults at the moment. So we're really looking at aging bats using things like CT scans of skulls, bone fusions, linear measurements, and also teeth, to try and see if we can't get a measurement in the field that's going to give us better data to understand age, because age will give us an idea of high-risk periods if we can narrow it down to a specific time period. And then movement patterns is very important in terms of where they feed. So we've had trackers on our fruit bats for quite a while, and we could track them at different times of the year. They feed in different areas. So if there's a lot of fig trees, they stay in the rural areas, they're away from the houses. As soon as it becomes winter months, they're in the yard of the people eating on their mangoes and on their other fruits. So immediately you have a time period of risk and when they're actually going to get into contact with humans. But they also do, they will drop the fruit or they will spit out the pits, and that can then lead to other animals eating it if there's saliva contamination of the fruit. So we're really trying to overlay this with our virological data to see risk. And one of the recent studies that one of my postdocs did, and she's a zoologist, is really looking at land use changes around some of our important bat caves in South Africa. And our caves are in protected areas. Most of our caves are, but there's still land use changes. And what was quite concerning is that in the past few years, there was a 10% increase in human activity closer to these caves. So we're moving into a situation where we're bringing together caves that was usually quite far away from people, directly into areas into closer contact with humans because of urbanization and people moving into these areas where their houses and their crops. So this is quite concerning from a conservation perspective, but also from a virology perspective. And this is just some of the work she's done. We overlay all of this with Human Modification Index, trying to use our census data and not global data, where we're also trying to see where's the most people, where is people living, what type of areas that they live in and their type of housing, to try and overlay the risk with our virology data. And then really looking at this bigger picture. So we also have human behavior studies currently going, coupled with serology and virological assays. And we don't know all those issues yet, but what we can see is that there's different issues in terms of human behavior in different areas and different cultures, even in South Africa, which complicates this whole situation. If we think back to COVID, we can say animal markets is a problem. Let's ban them worldwide. But the reason why there's an animal market in China is very different from an animal market in Western Africa. And if you don't understand those reasons and you're not really on the ground and investigating it, the type of actions that you will put in place to mitigate it will not work. So it's really going back to communities and really understanding what's the factors on the ground from a human behavior side that's leading to some of these actions and how we can mitigate it. We say sometimes regional, but I think regional is even too broad. It's really on a community level where we think there's risk. So that's in short the bigger picture and some of the work we've done. It's been funded by several agencies, including my research chair and US Threat Reduction Agency, and several institutions, both in South Africa and the US, has been involved in this work for several years. And if you want to contact me, you're welcome to email me or ask me questions afterwards. I will be happy to answer it. Thank you. Bye.
Yeah, thank you very much for that terrific talk, Professor Markotter. That was really awesome. So we will take some questions now. We will try our best to accommodate as many questions as we can. However, if your question does not get covered, please feel free to reach out to our speaker directly. Remember to submit your questions to Siang via the chat function. Let's see, and I think we have one question from Masa. So let me, Masa, if you want to unmute yourself.
Thank you.
Yeah.
Oh, thank you so much for your inspiring talk. And I'd like to ask last two slides. And I think these slides, I think you mentioned about the human and animal interface. And can you comment more on how we can attribute human influences to pathogen spillover among animals? I think you showed one slide, the human influence on pathogen spillover, but I'm interested in how we can attribute human influence to the pathogen spillover.
So I hope I hear you correctly. So you're asking how can we as humans contribute to that spillover interface? Is that what you're asking?
And, how can we attribute, like how can we associate, or how can we do, like how can we discover like more causal relationship from human influence to pathogen spillover among animals?
Yeah, so I think so this, let's take an example in South Africa. We know somebody tells us, oh, these caves are used for religious purposes. So we can just say, okay, that's the issue. Let's just shut down all religious purposes in caves. We're not gonna, people shouldn't go into caves. Or we just say, but that's not so easy. We really need to understand it. So we need to go to communities, and we usually do questionnaires and try and understand the interaction with bats and the interaction with caves and what's the activities that actually happen and what's the reasons why it happens. And then we can actually assess risk. A lot of times the risk is not that high. If you really start talking to people, no, they're actually not going into the cave. They're just standing in front of the cave, making a fire, and then you don't have to go that far as to say people shouldn't go into caves. And it's the same with the COVID situation. We decided very early in the outbreak, due to some information, that the animal markets are the reason. We have absolutely no scientific proof of that yet. So we should be very careful with the human behavior studies to, I think, make decisions on mitigation actions without scientific proof. And that's going to take time, because I can promise you, to try and figure out bushmeat in certain countries and what people actually eat, we can say, let's ban all bushmeat. But I can tell you now in South Africa, the type of bushmeat that we still eat is not that high risk. It's not bats, it's not rodents. Other countries, it's maybe higher risk. So we can't have these general rules without having the scientific evidence. And that scientific evidence is going to take time. And it needs to be very region and cultural specific. And a lot of people are pushing to move science back to community level. Get the communities involved from the beginning and work from that perspective, instead of just, from a top-down approach where we say, I've got a virus in a bat, now you will stop doing all of this. And really understanding the practices. I hope that answers your question. I hope I understood it correctly.
Thank you so much.
Awesome. I think we've got another question from Eva. So Eva, if you'd like to unmute.
Hi. Yeah, I'm interested in why bat viruses are considered like more dangerous or more likely to make zoonotic leaps than other animals. And like if bats are just the intermediate hosts that happen to be there, or if bats have some, receptors or other mechanisms that make the zoonotic leap more likely.
So I think the reason why bats have been implicated is there's been one or two good examples where a bat virus is actually spilled over to people. One of them is Marburg virus, where we have pretty good evidence, the virus is in the bats, the virus spilled over to humans. We could make that link with virology evidence. Nipah is another good example, and rabies has always been a good example. But then we basically extrapolated from that a lot of things that we assumed. I mean, if we go back to SARS coronavirus, I call it always SARS coronavirus 1, the first outbreak, we still don't have good evidence that bats was responsible for any human cases or spillover. We find related viruses and that's it. For years, it was just related viruses that couldn't even recognize human receptors. Then a few years back, we actually find a bat virus that could recognize a human receptor. It took us, I think, 10 years, 15 years to find that. It wasn't so easy to find that. With a lot of the other viruses, Ebola, we find viruses once in a study in bats. We could never link it to a human outbreak. So we know a lot of the recent Ebola work linked to bats that was linked to the Sierra Leone outbreak was all speculative. It was not based on virology data. It was because a bat was in the area or a kid was playing with a bat, no virology data. So I think it's sort of through the years, because there was this assumption that it was linked to bats, we assumed it without very good virology evidence. And it may still be true. It may still be that there's a lot of bat viruses that could potentially spill over. It may be that a bat was responsible for the original origin of SARS coronavirus 2, SARS coronavirus 1, Ebola, but the virology evidence is not that great. And I think that actually puts a responsibility on the scientific community to really investigate these things properly, because otherwise it leads to questions and speculation. And we really need the virology data. But I think it escalated from actually the SARS coronavirus 1 outbreak into a situation where we were blaming bats for everything, and with very few concrete virology data.
Awesome. I think actually Jason Blackard has a related question that they would like to ask.
Hi, Wanda. Thanks for the presentation. I actually work with some of your colleagues at UP in the Department of Medical Virology, so it's cool to hear your topic and your expertise. So a simple, well, I don't know if it's a simple question or not. Given the metaviromics data, and I'm going to call it that because it sounds like a nice word to me as a virologist, what percentage of the data that you look at, whether regardless of the species of the study, do you think is actually unclassified viruses? Like I'm trying to figure out how many truly unclassified new viruses you're identifying or can identify within these massive data sets that you generate.
I think it's still huge. We just went through an exercise like that to look for Paramyxoviruses, and so many of our reads are not host, and they sort of viruses, but they're not matching anything, and it's very difficult to actually decide where they belong. And metagenomics is not so straightforward with bat samples, and especially if you've got this high diversity. So I mean, there's people. ECHALT is actually one of the groups claiming that there's millions of viruses out there that we haven't discovered, which is great. But my perspective has always been, is that really where most of the money and the focus should go? Yes, we should know. On high-risk interfaces, I think we should know the diversity, but I think even as a virologist, I would say that the focus needs to shift more on the interface that's going to lead to spillover. So we know there's a diversity. Is it going to help us to know there's 1 million different viruses or 1,000 different viruses?
Yeah, good.
And we need to know, but I think our focus, and that's also where the OLEP panel is going, that I'm co-chairing, is really looking at coming back to One Health, which has been quite an old definition, but really looking at a more holistic picture, even if we do virology. But there's high diversity out there. We sometimes can't even classify them.
Thank you.
Thanks. So Bill, I think you've got a question.
Thanks. Thanks, Brad. And also thank you for the talk. It was really, it was great. I was just going to add to the thing you just said about diversity. The only place where I think I would disagree is that if by recognizing there's a huge amount of diversity, it helps us understand that if we do find an emerging virus, we shouldn't expect there to necessarily be a close match that we already know about, because there is just so much diversity. Because I'm saying that as having reviewed certain books and found them quite frustrating. The question I wanted to ask you, though, was, have you done any serology going into places which are in proximity to bat caves and looking to see if populations in close proximity to them are more likely to have antibodies to the viruses which are circulating, just as a proxy for the amount of contact between the populations?
Yeah, so we've done before with the tools that we had, and we would get, matches, but it's extremely difficult if you don't have a control population to interpret these things. So we never published it. So what we've recently done is we've implemented better serology in South Africa that's really more focused on the African diversity. Because with coronaviruses, it will just react with everything. So you anyway have an issue. But with the phylos, we now have better serology, and with the paramyxes also. So we're starting to look at that, and we can see certain trends in high-risk groups. So we have a group of bat researchers that we test regularly after they've been into different caves, and we're trying to look over time. We now have a group that's linked to some of the religious activities in the community. So serology, I think it's got value to sort of give you a broad idea of where you need to look, but you also need to be extremely careful how you interpret it. And there's a lot of cross-reaction. There's been studies published in Africa that implicated pigs and other animals based on one serological positive. So we also need to be really careful where we directing risk if we don't have a proper study and a lot of serological positives, and we know we don't have cross-reaction with some of the other viruses. But I think it's got a lot of value in combination with other studies. So we're looking at that, but it's complicated to interpret.
Yeah, right. Interdisciplinary, as you were saying.
Yes.
Cool. We're getting close to the top of the hour, but I think Siang has another question.
Yeah. Hi. Thank you so much for this wonderful talk. I'm curious, we know some, like you mentioned at the beginning, we know some spillover events happening through a vector species. For example, like a mosquito or a tick or some mostly like insects. So I'm wondering if you could comment more on how we can identify this vector mediated spillover event, and how we can identify the key vectors in this type of things. Thanks.
So if you're talking about vector-borne specifically, it's probably the same situation as bats. We're testing bats, we're finding a positive, but what's the real risk of spillover? Is that a incidental detection, or is it actually something that's maintained? And I think it again goes back to longitudinal surveillance. I'm very for doing surveillance over time, over seasons, in one place, which is extremely time consuming. It is expensive. You need to basically be in country to do that. But that starts giving you better answers about what role that vector can maybe play. And then your experimental infections also answer some of those questions. And in terms of vectors, especially your vector competence studies, which is really trying to look at if they can maintain the viruses. And I know people are always not so for experimental infections, but if you have a very specific question, those type of things really add a lot of value if you plan them right to really understand in terms of vector transmissions, but also in terms of mammal hosts on how the virus can potentially be transmitted and maintained. But there's very few labs that can still do that with all the biosecurity and biosafety.
Awesome. So yeah, we are at the top of the hour, so I'm going to close this out. On behalf of the Center for Communicable Disease Dynamics and the Harvard T.H. Chan School of Public Health, let's thank Professor Markotter again for joining us today and for a fantastic talk and discussion. Please do get in touch with any of the seminar organizers if you have questions for us or our speaker. I know I've got some questions, so expect an email from me. And also check out the CCDD website to stay up to date on upcoming seminars and other events. Next week's seminar will be given by Professor Andrew Reed from Penn State University, and he will be talking about pathogen evolution in a vaccinated world. So thank you again for joining. Have a good day. Bye.
Hi, thanks everyone. Bye.